Thank you very much. Can you all hear me? Thank you. Thank you so much for having me here today to talk about some of my work. I'm really thrilled to be here. It's in fact almost the first time, except for VOM last year, when I give a talk to actually some people who work in phage, as opposed to clinicians or veterinarians. So this is just some of the work we do, and I'll show you a little bit about my group at the end if I get time. Right. Does it move? It doesn't move. It doesn't move, sorry. It started. Yeah, okay. All right, so why are we interested? I work in a veterinary school at the moment. I've been working for many years with clinicians at the Westmead Institute for Medical Research. That's where I started to do my work on bacteriophages. But since 2024, I've moved to a vet school. And so we've been looking at interventions potentially in the animal setting. Companion animals are extremely important. In Australia, a lot of people have pets and they care about their pets. So we have sort of a huge field here to explore where we could really look into potential application of phages for therapeutic treatment. Why hard to treat UTIs? Because like in people, dogs and cats, particularly old ones, can get these infections, and they are in fact frequently diagnosed, particularly when we are looking at older animals and animals with maybe some pre-existing condition. You can have sporadic UTIs episodes. They tend to sort of resolve pretty quickly by themselves, but we have also recurrent and chronic UTI in these companion animals, and these are more than 3 episodes of infections in 1 year. These infections are often treated empirically. We're talking about vet settings here. We don't do really the kind of surveillance and kind of preparation for treatment that we do for people. And we have different treatments recommended in sporadic or recurrent cases, but really the first line of the first choice is first-line antimicrobials like amoxicillin or trimethoprim, often with sulfonamides. And this is in line with the international guidelines. We didn't really know what we were dealing with, though, in terms of the bacteria that cause UTIs in the vet setting in New South Wales, in Sydney in particular. So we had a student, student, Annie, who sort of did this study looking at the epidemiology of these infections in a large collection that is coming from the diagnostic lab just next door to the vet school. And, or it's actually within the vet school. And what we find out, well, we had this huge collection of about 1,500 isolates from 2012 to 2021, and it's continuously increasing. And Annie looked at the data from the diagnostic lab, which is ID for the bacteria isolated from these cases, plus antimicrobial susceptibility testing data. And not surprisingly, we found that 58% of cases were associated with E. coli. Some enterococci, some staphylococci, but predominantly E. coli, and highest rates of multi-drug resistance were again in E. coli. And overwhelmingly, cases of multi-drug resistance were linked with recurrent UTIs. So here's a space where we can think about using phages. So to start this study, I selected a subset of multi-drug resistant E. coli from this collection from 2012 to 2021. And we added also another 41 from, well, what it turned out not to be non-multi-drug resistant isolates, current ones after COVID. Crucially, we stopped sampling during the COVID years. And we have the same sort of roughly the same amount for cats and dogs. So that's always good. Importantly, what we want to do to see if we can apply phages for therapy, I think it's very important to know what you're dealing with. So we did the genomic analysis of the multi-drug resistant population and also non-multi-drug resistant, but I'm showing you here only the results for the multi-drug resistant subset. And you can see that I mean, obviously, this is not great phylogeny. Phylogenists out there, don't kill me. It's a handful of samples, but we really wanted to know whether we had clonal expansion, some key sequence types that were coming up all the time. It's not the case. So that's a bit tricky for application of therapy, but it's important to know. And you can see that we have sort of three main clades. I can't find my pointer here. Or four main clades, sorry, however, where isolates are sort of clustering more or less. And importantly, we found in this collection sequence types that are clinically relevant also in human infections, not just for UTIs, but also for other extraintestinal infections like ST38, ST131, and ST73. When we look at the profiles here of our population in terms of antimicrobial resistance, these are only the feline isolates. You can see that we have, when we have multi-drug resistant isolates, we have resistance to a range of classes of antimicrobials. Look at the bottom set, that's the most recent set after COVID, and we hardly find the resistance. This will probably change if we keep testing our newest isolates. All right, so there is a method to the madness, like for all of us. While working at Westmead, the first years that I was starting to get my head around phages for therapeutic applications, I sort of came up with a protocol that I try to follow when I'm trying to design therapy. I'm not an interventionist, I'm not a clinician, I'm not a vet, I'm a microbiologist, so I look at the full picture. Important is to look at your base or your target population, I think, and then look at the interactions between phages and bacteria. So now I'm going to give you a messy overview of the bits and pieces or all the aspects that we're trying to look into before we think about intervening and doing our trials, because this is, in fact, the ultimate aim to actually intervene in sick animals. Most likely it will be dogs. So we started off by trying to find phages for this collection, and we focus mainly on not on the singletons at the bottom, but on sort of the main clades, let's call them. And we isolated and have characterized approximately 34 phages de novo, and you can see that for most of the top clade, we find phages. That's the purplish sort of color. I am blinding my old age, so if there is a pointer, the arrow, I can't see it anymore. Anyhow, in purple there, you have the hits. So about 34 phages. We had a much larger collection that we can access for XPEC in general, not kind of UPEC, which is the uropathogenic E. coli, but phages that were isolated previously, again, extraintestinal pathogenic E. coli. And these were tested also against this population. We chose single isolates, can do everything on everything. So for sort of representatives of main sequence types that might be pathogenic and pathogenic in humans and animals, and representative of these sort of clades. What did we find? We found that the phages we isolated had cross-reactivity against different E. coli types and E. coli isolates. And crucially, when we put them together, starting sort of to mix them in cocktails, we found that cocktails behaved better than single phages, not a surprise. And these are just highlighted the key isolates that we keep working with or focusing on. And what I wanted to show you also in these very colorful slides from my students is that phages that were isolated against animal UPEC also work against human isolates. Sorry. Oh, this is the one. Against the human, pardon me, that's right. So phages isolated against the animals also work on human isolates. So extraintestinal pathogenic E. coli coming from sepsis and UTIs in humans, and that phages isolated against human isolates work for our animal isolates. So we can look at our full set of phages to think about intervention. Also, importantly, the phages that we isolated against the multi-drug resistant E. coli work also against the current or most recent isolates that are not multi-drug resistant. We test activity in liquid as well as in on agar, of course, and we find that generally the cocktails behave well in liquid as well. So this is sort of standard. What has been of interest and that we've focused on a little bit more recently, is multimodal combination using phages along with some other antimicrobial. We've started off looking at the common antimicrobials that we use in treatment. So amoxicillin, ampicillins, amoxicillin clavulanic acid, and trimethoprim. And we can see that, of course, the picture varies for the different isolates and different phages. And for example, in this case of DCC2 isolates, we see that, for example, phages alone work very well against the multi-drug resistant isolates. Combinations don't really give us a better hit, but we can see that, for example, for phage 3, which is a bit of a dud, if we mix it with the beta-lactam, actually we have a reduction in bacterial load, which is, hopeful and interesting. If we look at another isolate, DCC14 is an ST131, so a pandemic clone circulating everywhere in humans and animals in various infections. We can see that a phage that doesn't work very well combined with antibiotics reduces, we have a significant reduction of bacterial load with the 3 antibiotics, trimethoprim as well, which is not a beta-lactam, which is interesting. So looking at how this adaptation or this synergy occurs molecularly will be very interesting. We have also looked at combinations with antimicrobial peptides. We luckily am working with a very talented veterinarian, Kate Worthing, who in a previous life in the US as a vet, she actually isolated a feline antimicrobial peptide from cat saliva, which has very good antimicrobial activity. And we wanted to test this, it's produced by Staphylococcus species. And we wanted to test whether this antimicrobial peptide worked with our phages and alone against our E. coli's. I'm not going to take you through the details. You see that I'm not, but what we do is we combine antibiotics and antimicrobial peptides at different concentrations, and then we have our dutiful controls. And what's important to see is how long in these assays, I should have said it before, but I'm sure you've all seen them before, how long bacterial growth is suppressed compared to the control. So here you can see, don't worry about the details, but that the line, when we have cocktails or mixes of phages combined with antimicrobial peptide, we have growth suppression for longer. And so this is kind of a positive result, but as I'm going to show you now, the picture is pretty varied. And for each isolate and each antimicrobial peptide phage concentration, there is a sweet combination. So in this case, cocktail number 3, the phage combination number 3, at an MOI of 100 with the AMP at 100% concentration works better. In other cases, we've seen better results with AMP concentration at 25%. Because UTI associated E. coli is great at making biofilms, we wanted to also test this because it's very important when we think about intervention in chronic and recurrent cases. Biofilm formation is a real possibility. Cats and dogs get catheters as well. And so we have started looking on key isolates at the production ability of biofilm in both liquid media and on catheters. And we saw that these E. coli produce in general, and these are very different isolates, biofilms better in urine, wow, minimal medium, than in our lab media. And that catheters are very great surfaces for producing these biofilms, particularly in urine. So this is definitely something that we need to take into account when we will think about intervention. We are also looking at the activity of the antimicrobial peptides in these biofilm assays for biofilm prevention and biofilm disruption. We have a bit of data just for biofilm prevention, and the phages and antimicrobial peptides alone didn't give much joy. But when we tried a different concentration of the antimicrobial peptide and the phage, so we modulated that, we saw very good preliminary results. These need to be repeated. They are very new, sorry. Also, E. coli associated with urinary tract infections and extraintestinal pathogenic E. coli can enter eukaryotic cells. So we have to do also a little bit of work on seeing whether our phages can reach and then kill E. coli that have been internalized by eukaryotic cells. We have done this in epithelial bladder cells of humans last year, but I managed to get a bit more fun, and we got some promising results in the sense that, again, the interaction is based on the phage isolate pair, can be quite unique, but there are some hopeful results for at least preventing adhesion when the phage is present. A little bit more mixed results for whether the phages can really get rid of intracellular bugs. We are doing this now, though I got a bit more money, in feline and canine cell lines. So these assays will be, this is very much work in progress, and it will all be done diligently on the phages that we are now sort of narrowing our interest towards for trials. Okay. This was a really quick overview. I'm happy to talk to you later on anything we do. This is the group that we have set up. It's actually a network. It's not just my group of collaborators within the vet school, and plenty of students who help a lot. We have a web page on the UCID site. It just gives you an outline of all the projects we're involved in, because we work on top priority pathogens, not just E. coli, but a few that are of interest in animals and humans. The focus is One Health. And not only in Australia, but we work a lot with Southeast Asia as well, particularly India and Vietnam. And this is a bit a collection of phages, just to give you an idea of why I'm so muddled about some things, because we have tried to sort of look at potential interventions in these past few years, and now we are running into some real sort of areas of focus where we might have translational or opportunity to test phages more readily than in others. I don't work only on gram negatives, but I love gram negatives. I do have to work on some gram positives. So they are there at the bottom. And thank you very much for listening.